Dear students, very warm welcome to all of you. Today we are here to discuss about meiosis. Well, the word meiosis has been derived from Greek meion, means to diminish. It is a type of cell division in which a nucleus divides into 4 daughter nuclei, each containing half the chromosome number of the parent nucleus. Meiosis occurs in all sexually reproducing organisms in which haploid gametes or spores are produced. Meiosis involves 2 consecutive divisions of the nucleus and leads to the production of reproductive cells, that is, gametes in animals, and to the formation of spores in plants, fungi and most algae. Meiosis begins when the chromosomes, which have already duplicated, condense along the center of the nucleus, and pairs of the homologous chromosomes undergo crossing over, whereby some of their genetic material is exchanged. The pairs of chromosomes then separate and move to opposite ends of the cell, and the cell itself divides into 2 cells. In the second stage, each of these 2 cells also divides into 2 cells. Meiosis thus produces 4 cells, each of which contains half the number of chromosomes as the original cell. Some or all of the 4 cells may become functional gametes or spores. Meiosis was discovered and described for the first time in sea urchin eggs in 1876 by noted German biologist Oskar Hartwig. It was described again in 1883 at the level of chromosomes by Belgian zoologist Edward van Benden in Ascaris worms eggs. Meiosis is thought to have appeared 1.4 billion years ago. The only supergroup of eukaryotes which does not have meiosis in all organisms is excavata. Well, during meiosis, the genome of a diploid germ cell, which is composed of long segments of DNA packaged into chromosomes, undergo DNA replication, followed by 2 rounds of division, resulting in 4 haploid cells. Each of these cells contains 1 complete set of chromosomes, or half of the genetic content of the original cell. Dear students, let us take up the stages of meiosis. Meiosis can be broadly divided into 2 major phases, that is, meiosis I and meiosis II. Let us take up meiosis I first. During meiosis I, homologous chromosomes separate, producing 2 haploid cells. So, meiosis I is referred to as a reductional division. In meiosis II, which is an equational division similar to mitosis, which occur whereby the sister chromatids are finally split, creating a total of 4 haploid cells per daughter cell from the first division. Meiosis I can be further subdivided into following phases. Prophase I. During Prophase I, DNA is exchanged between homologous chromosomes in a process called homologous recombination. This often results in chromosomal crossover. At this stage, non-sister chromatids may cross over at points called chiasmata. Prophase I comprises of following subphases, that is Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. Now, in Leptotene, which constitutes the first division of Prophase I, it is also known as leptonema. The word is Greek one, meaning thin threads. During this stage, individual chromosomes begin to condense into long strands within the nucleus. However, the two sister chromatids are still so tightly bound that they are indistinguishable from one another. Second one is zygotene. The zygotene stage is also known as zygonema. The word again is Greek one, meaning paired threads. During this stage, the chromosomes approximately line up with each other into homologous chromosomes. This is called the bouquet stage because of the way the telomeres cluster at one end of the nucleus. At this stage, the synapses, that is pairing or coming together of homologous chromosomes, takes place. Now we have one more stage which we call Pachytene. The Pachytene stage, also known as pachynema, from Greek words meaning thick threads, contains the chromosomal crossover. Non-sister chromatids of homologous chromosomes randomly exchange segments of genetic information or regions of homology. However, sex chromosomes are not wholly identical and only exchange information over a small region of homology. Exchange takes place at sites where recombination nodules, the chiasmata, have formed. The exchange of information between the non-sister chromatids results in a recombination of information. Each chromosome has the complete set of information it had before, and there are no gaps formed as a result of the process. Because the chromosomes cannot be distinguished in the synaptonemal complex, the actual act of crossing over is not perceivable through the microscope. Now we have one more stage, Diplotene. The Diplotene stage is also known as diplonema, from the Greek words meaning two threads. The synaptonemal complex degrades and homologous chromosomes separate from one another a little. The chromosomes themselves uncoil a bit, allowing some transcription of DNA. However, the chromosomes, the homologous chromosomes of each bivalent remain tightly bound at the chiasmata, the regions where crossing over occurred. Now, diakinesis stage. The word diakinesis is a Greek one meaning moving through. During this stage, chromosomes condense further. This is the first point in meiosis, where the 4 parts of the tetrads are actually visible. Sites of crossing over entangle together, effectively overlapping, making chiasmata clearly visible. The nucleoli disappear and the nuclear membrane disintegrates into vesicles and the meiotic spindle begins to form. During synchronous processes, 2 centrosomes containing a pair of centrioles in animal cells migrate to the 2 poles of the cell. These centrosomes, which were duplicated during S phase, function as microtubule organizing centers, nucleating microtubules, which are essentially cellular ropes and poles. The microtubules invade the nuclear region after the nuclear envelope disintegrates, attaching to the chromosomes at the kinetochore. The kinetochore functions as a motor, pulling the chromosomes along the attached microtubule toward the originating centriole, like a train on a track. There are 4 kinetochores on each tetrad, but the pair of kinetochores on each sister chromatid fuses and functions as a unit during meiosis I. Microtubules that attach to the kinetochores are known as kinetochore microtubules. Other microtubules will interact with microtubules from the opposite centriole are called non-kinetochore microtubules or polar microtubules. A third type of microtubules, the ester microtubules, radiates from the centrosome into the cytoplasm or contacts components of the membrane skeleton. Now, metaphase I. Homologous pairs move together along the metaphase plate. As kinetochore microtubules from both centrioles attach to their respective kinetochores, the homologous chromosomes align along an equatorial plane that bisects the spindle due to continuous counterbalancing forces exerted on the bivalents by the microtubules emanating from the 2 kinetochores of homologous chromosomes. The physical basis of the independent assortment of chromosomes is the random orientation of each bivalent along the metaphase plate with respect to the orientation of other bivalents along the same equatorial line. Now, anaphase I. Kinetochore microtubules shorten, severing the recombination nodules and pulling homologous chromosomes apart. Since each chromosome has only one functional unit of a pair of kinetochores, whole chromosomes are pulled toward opposing poles, forming two haploid sets. Each chromosome still contains a pair of sister chromatids. Non-kinetochore microtubules lengthen, pushing the centrioles farther apart. The cell elongates in preparation for division down the center. Now we have one more stage, which is telophase-I. The last meiotic division effectively ends when the chromosomes arrive at the poles. Each daughter cell now has half the number of chromosomes, but each chromosome consists of a pair of chromatids. The microtubules that make up the spindle network disappear, and a new nuclear membrane surrounds each haploid cell. The chromosomes uncoil back to chromatin. Cytokinesis, the pinching of the cell membrane in animal cells, or the formation of the cell wall in plant cells occurs, completing the creation of two daughter cells. Sister chromatids remain attached during telophase-I. Cells may enter a period of rest known as interkinesis or interphase-II. No DNA replication occurs during this stage. Dear students, let us discuss meiosis II. Meiosis II is the second part of meiotic process. Much of the process is similar to mitosis. The end result is the production of 4 haploid cells from the 2 haploid cells produced in meiosis I. The 4 main steps of meiosis II are Prophase II, Metaphase-II, Anaphase-II, and Telophase-II. In Prophase-II, the disappearance of the nucleoli and the nuclear envelope again, as well as the shortening and thickening of chromatids takes place. Centrioles move to the polar regions and arrange spindle fibers for the second meiotic division. In Metaphase-II, the centromeres contain 2 kinetochores that attach to the spindle fibers from the centrosomes at each pole. The new equatorial metaphase plate is rotated by 90 degrees when compared to the meiosis I, perpendicular to the previous plate. In Anaphase-II, the centromeres are cleaved and allowing microtubules attached to the kinetochores to pull the sister chromatids apart. The sister chromatids, by convention, are now called sister chromosomes as they move toward opposing poles. The process ends with telophase-II, which is similar to telophase-I and is marked by uncoiling and lengthening of the chromosomes and the disappearance of the spindle. Nuclear envelopes reform and cleavage or the cell wall formation eventually produces a total of 4 daughter cells, each with a haploid set of chromosomes. Meiosis is now complete and ends up with 4 new daughter cells. Now let us take up significance of meiosis. Meiosis facilitates stable sexual production. Without halving of ploidy or chromosome count, fertilization would result in zygotes that have twice the number of chromosomes as the zygotes from the previous generation. Successive generations would have an exponential increase in chromosome count. In organisms that are normally diploid, polyploidy, the state of having 3 or more sets of chromosomes, results in extreme developmental abnormalities or lethality. Polyploidy is poorly tolerated in most animal species. Plants, however, regularly produce fertile, viable polyploids. Polyploidy has been implicated as an important mechanism in plant speciation. Most importantly, recombination and independent assortment of homologous chromosomes allow for a greater diversity of genotypes in the population. This produces genetic variation in gametes that promote genetic and phenotypic variation in a population of offspring. Dear students, this is all about meiosis. In this topic, we have learnt about meiosis and its various stages, genetic recombination, and significance of meiosis. Thank you very much. God bless you.